Namaskar. Today we will study cryopreservation. So cryopreservation is a preservation technique where we can preserve the biological specimens like living tissue, cell or cell line. So how we can preserve that? We need a temperature closer to minus 200 degrees Celsius. Exactly minus 196 degrees Celsius is the temperature required for cryopreservation. And for that we need something which can provide that temperature, and that is liquid nitrogen. And the container in which we keep the biological specimen is called liquid nitrogen container. So there is a temperature controlled storage area where the temperature is marked here, and the biological specimen is kept inside this container, and surrounding it there is the liquid nitrogen envelope. So this liquid nitrogen will provide, here the liquid nitrogen is there, so this liquid nitrogen will provide the temperature, and outside there is vacuum insulated outer layer, the jacketing, vacuum insulated jacketing is there outside, and here inside this is called the temperature controlled storage area where the biological specimens are kept. So why we use liquid nitrogen? Because the boiling point of liquid nitrogen is minus 195 degrees Celsius, and at this temperature living cells remain live outside the body, so in vitro they remain live. The enzymes, the cellular process, the activity, all are halted at this temperature. There is no apoptosis or cell death at this temperature. There is no formation of water crystals, because the water crystals may burst the cell. So there is no formation of water crystals at this temperature. So for cryopreservation we need the temperature around 200 degrees Celsius. So what is the process? So process is first we need to, before confluence we need to passage the cells. So say before it reaches the 90% confluency, we have to passage the cells, to suspend them in a serum containing media, centrifuge, aspirate the supernatant, resuspend the cells in 10% dimethyl sulfoxide in fetal calf serum. Then we need to transfer to a cryovial, freeze it at minus 80 degrees Celsius. Then finally we need to transfer to liquid nitrogen storage tank, and the temperature is set at minus 196 degrees Celsius. When we want to use those biological samples, again we have to return the temperature to room temperature, and that process is called vitrification. So vitrification is a process of step by step increment of temperature. So directly we cannot change the temperature from minus 196 to 35 or 37 degrees Celsius, the room temperature. We need to do it step by step increase. So step by step increment of temperature from minus 196 or minus 195 degrees Celsius to the room temperature, that is 37 degrees Celsius or 35 degrees Celsius, that is called vitrification. So from minus 196 degrees Celsius we need to change it to minus 100, then to minus 50, then to 0 degree, then to 25, then to 35 degrees Celsius. So this is the process called vitrification. So what are the advantages of cryopreservation? Cryopreservation reduces the risk of microbial contamination. It reduces the risk of cross contamination with other cell lines. It reduces genetic drift, and it also reduces the morphological changes. So this is all about cryopreservation. Thank you.